So welcome back. I hope you had some refreshments and some nice break. So the second part, you will hear a bit less from me because you might be by now fed up by listening to me. So we will meet together for the first hour, and then I will leave the floor to my colleague Gio. Then there are two things that I would like to mention in this session, which is titled The Real World Challenges. So the first thing is some glimpse on what could be done with the genetic and genomic data. Genomic epidemiology has exploded in the past few years thanks to really a change in the way technology works when it comes to sequencing. We went from really highly costly approaches to sequencing that made large-scale sequencing impossible until a few years ago. While during the pandemic, we all heard a lot about genomic data, especially to track the rising and resurgence of different variants of the pathogen from COVID-19, to really try and see how different variants could have different symptomatology or different transmissibility, or even escape, for example, the vaccination measures that have been developed for the first version, say, of COVID-19. So genomic epidemiology is really useful to somehow monitor trends, both at the national and local level, and how different strains, different lineages of the pathogen can emerge due to mutations, both through shifts and really changes in the way mostly the antigens that the pathogens use to invade the immune system. But also they are super useful to investigate, for example, clusters, to understand whether a cluster of, so a group of infections are actually a cluster from the same outbreak, or if they are from different outbreaks, or if cases have been imported by different areas with different strains, and so on and so forth. And I would just like to cite a few works that have been using genomic data to couple with modeling. In particular, there is a work done recently by the team of Vittoria Polizza and Chiara Poletto in collaboration with Philippe Lemay in France, and they really used the combination of different genomic data for influenza to really track how different strains of influenza spread around the world, and how can these be used to inform global epidemic spreading modeling for influenza around the world, and really to differentiate how different strains circulate on the northern hemisphere, southern hemisphere, or even different countries, and how this has an impact on the epidemic in that specific country. And similarly, they did some work for the respiratory syncytial virus and the genomic data to really look at how evolution of the sequences of RSV can be captured also through modeling in the very same way that was done for SARS-CoV-2. So I won't go into the details of everything that has been done also for COVID, but there has been a lot of use for genomic data from COVID to really study the invasion of different variants from a geographical perspective. So the combination of genomic data with spatial spreading is super interesting, and we will hear a lot more about this from Mattia in the next session. On a smaller scale, genomic data can also be used to represent outbreaks. So this is something very, you cannot see it from the title very well. So you can, for example, reconstruct chains of transmission in closed settings such as schools or households based on genomic data, contact data, and surveillance data. And this is a work done again by Andrea Tornelli in collaboration with INSERM and Vittoria Polizza, where they used data in the primary school, also in combination with genomic data, so sequencing of the samples taken from the children in school that were presenting symptoms to reconstruct the chains of transmission in this school. So this is just to give you a glimpse of what can be done with the genomic data in combination with modeling. And you will hear a bit more about this when you will hear about the contact networks and the mobility talks. The last part of my presentation is on real-world challenges, and it's really a way to show from an operational perspective what can be done when these modeling tools are applied in real-world context. In the past 2 decades have seen really the rise of so many emergent new diseases or recurring diseases. We had H1N1, 2009 H1N1 influenza pandemic, which was at some point became a mild influenza epidemic, but until everybody was sure that it was a mild influenza epidemic, there was a lot of uncertainty on what would happen, when the peak would come, and when the epidemic will invade the population. And then we had several others, large-scale public health emergencies. In 2014, the Ebola situation in Western Africa, the Zika epidemic in South America that also became a problem in North America, and then of course COVID-19. And I will show you very briefly what the data-driven simulation framework that also relies on big data could use in this context. And you have already seen, this is a model that was developed by our president, Spignani, in collaboration between ISI and his lab, the MOX lab at Northeastern University. And I will just give you some more details of what you have seen, with respect to what you have seen before of the model. And as I was saying, it's a model that comprises different data sources, different modeling approaches. It includes high-resolution demographic data, mobility data, and also epidemiological data, in a framework that somehow simulates 8 billion of individuals moving around the world and interacting with each other to create outputs that are highly realistic and can be used to really inform operational decisions during pandemic crisis. This is called the Global Epidemic and Mobility Model, and it's quite a flexible approach that has now been used in several of the contexts that I just mentioned. The main ingredient is a high-resolution, detailed representation of the density of the population around the world, and it's based on open data from NASA, actually, and it's possible based on the luminosity as seen from satellites. In a certain geographical area, you can infer how many people live there based on how much light is emitted in a certain area. And so you can estimate with a geographical grid covering the whole world with say dimensions that are more or less 25 kilometers by 25, to know how many people live in each cell of this global grid based on this open data provided by NASA. And why is this important? Because we know not only how many people live in a certain region with such a high resolution, but we also know how many people travel from one place to the other. And the idea is that to have a realistic network of mobility, you can couple each geographical region through short range or long range mobility based, for example, on where people live. What Vespignani's team did was to somehow arrange these geographical cells around airport basins, for example. If you live not far from Bangkok, it's very likely that to go to another country, you will take a flight from the city. So you will travel to the city and then take a flight to London, for example. But if you live in Vietnam, it's very likely that you will come to Thailand to take a flight. You will take the flight from the closest big airport that is the nearest to where you live. And so the idea is to divide the whole world in airport basins that are coupled to each geographical cell that is the nearest to the airport from which a flight can be taken. And this is true also for the short range mobility. If you have to take a train, it's unlikely that you will go to Chiang Mai to take a train. You will start from Bangkok, take a train from Bangkok, and go somewhere else. And so each geographical cell is coupled to the nearest, let's say, urban pole or reference, and then from there the coupling in terms of mobility is implemented. And this creates a very powerful set of layers of data for population, short-range mobility, and long-range mobility, on top of which a compartmental model can be simulated. In the very same way with the metapopulation approach that I mentioned earlier. You have geographical areas in which people mix in a, let's say, SIR fashion with a compartmental model describing this dynamic of infection. And then susceptible, infected, and recovered individual can travel based on the amount of people that live in a certain area, and also based on the network of short-range and long-range mobility. And this means that you have patches, geographical patches, coupled by mobility, in which the dynamics of the epidemic can be simulated. This means that you have several, let's say, different moments in which the disease is evolving. You have the travel of individuals and the dynamics of the disease, and they are all described by stochastic differential equations with the transport operators that couple geographical regions. But the ultimate parallel is your SIR on networks model that you have seen at the beginning. This approach can be really powerful and create a high resolution risk max for the importation of cases for a certain pandemic that, for example, can be simulated to start in a certain region with a certain transmissibility and then let be evolved on a global scale. Also from not only a representation that shows how many flights will carry infected individuals in how many countries, but also from a topological perspective, so really looking at the network, what are the nodes that are going to be infected first, because they are the closest on the topology of the network, as you can see on this representation here. And this is a tool that has been used first to predict the spreading of first to be tested on historical data for SARS-CoV back in 2002. And then it has been tested prospectively to really predict what will happen for H1N1, the 2009 pandemic, where there was really a real-time effort to forecast the peak of the epidemic. This was done throughout the summer of 2009, based on data collected in the first phases between end of April, end of June, more or less, when the disease became, from public health emergency affecting a few countries, it became a pandemic. Back at the time, the definition of a pandemic was based on the number of countries that were infected by the disease. That's the definition. That used to be the definition of the WHO. And so, based on this data and on the projections for the upcoming months, it was possible to predict the peaks of the epidemic in several countries around the world. And so this was based on data for mobility that were built in the model, but also surveillance data that were generated by each country. And this was very interesting because the H1N1 pandemic was the very first pandemic for which the first cases imported in a country were publicly announced on news outlets, on the media, on the internet. So the first case of influenza H1N1 2009 has arrived in Italy. The first case has arrived in Spain. And so thanks to this high-resolution information, it was possible to initiate the simulations and project what would happen in the following months. And the interesting part is that from a computational perspective, this is let's say, an agnostic approach that can be used not only for pandemics but also for many other epidemics. And the idea is that you sample the phase space from the parameters of your simulations, how much a disease is transmissible, what is the generation time, the time it takes to be recovered. All these are parameters that have to be inferred from the data, and from which you generate a set of trajectories, for example, and then you use the surveillance data that are continuously generated throughout the epidemic to somehow choose which trajectories simulated by the system best fit the data that you observe. This was very useful throughout the 2009 pandemic because it allowed us to really show that the peak of the pandemic could be reached in November. And already in summer it was clear that it was an influenza. It was not so different than seasonal influenza in terms of symptoms, in terms of transmissibility. But the normal epidemic for influenza would have peaked in February or even March, while it was shown some months in advance that this influenza would have peaked in more or less November, December. And the error that the projections provided was estimated by a few weeks. So it was a really precise projections that was then validated against real data, and it was done in real time with the surveillance data coming in the very first months of the pandemic. A similar approach, even though not including global travel, was then proposed for the 2014 outbreak of Ebola in Western Africa. Here, the crucial ingredient in the estimations for the projected cases was the kind of interventions. So here you see some projections of the number of cases for interventions that were including Ebola transmission units implemented on the ground and safe burial sites. So what the researchers found was that one of the main paths of transmission for Ebola was the burial procedures. And by somehow reducing the burial procedures that were taking place for people who were diseased by Ebola could really limit and contain the spreading of the disease. And this was a very crucial ingredient that would somehow account for social mixing in a way that could not really be foreseen before the epidemic. And the same goes for Zika. A very similar approach was used when it comes to global travel from across the whole American continent. This was the model that was used in this work in EMS by Spignani. And here, the crucial ingredient was the exposure to the vector. So they used high-resolution data about the distribution of mosquitoes in the Americas, coupled with the exposure driven by socioeconomic factors. So people who are more wealthy, let's say, live more in air-conditioned social context, while people who are less wealthy live more outdoor, and so are more exposed to the vector. And so accounting for these socioeconomic factors in South America led to the creation of very accurate projections, as you can see from this plot, in terms of the number of cases observed in a certain country affected by Zika. The last thing I want to mention is this work that came out in Science based on the projections for exportation of COVID-19 cases from China to estimate the impact of reduction of human mobility and really lockdown that happened in China. But the most, let's say, interesting part came when the same approach was used to study cryptic transmission. And so basically, thanks to this work, it was possible to show that when China went into lockdown, it was already probably too late to contain the spreading of the disease. Because if you look at how the importation of cases happened throughout Europe, was mainly driven by cases that were already present in the continent. Because there has been a huge phase of cryptic transmission not detected by surveillance systems, due to the fact that on one hand, COVID symptoms at the beginning were, let's say, confused with a normal, let's say, influenza, but also because the fraction of asymptomatic individuals for COVID is way higher than for other infectious diseases. And so the disease could spread, let's say, undetected for months before the first more severe cases related to pneumonia alerted the general community. And so when China went into lockdown, it was probably too late. And in Italy, if you think that we went into lockdown at the beginning of March, and the first cases were supposed to be detected in February, it means that the circulation of the disease was already super widespread, even at the end of December, beginning of January. I would like to conclude with the next frontier. What's next for modeling infectious diseases? Where this is a very exciting time. We have artificial intelligence coming for us, for the good or for the bad. This is a paper that came out a few weeks ago, I would say, in which the authors have tried to somehow provide a review of the new initiatives that artificial intelligence can provide in terms of modeling infectious disease opportunities. I invite you to read this paper. We don't have time to discuss it in details, but this is just to say that modeling infectious diseases is really a field in which innovation from data science, computer science, but also artificial intelligence will bring a lot of exciting things. And with this, I will conclude. And I don't know if we can take questions now, or maybe we'll do it at the end or later. So I will thank you and look forward to my group. Thanks.
Good morning. I'm very pleased to be here. Thank you again for the organizers for such an example. It's really exciting to spend time with you and share ideas and problems to involve. Let's see if this works. think we're earlier, so now.
Can you hear me well? Perfect. Okay, oh, that's my PC desktop. Sorry. Okay, perfect. What I want to share with you now for the remaining 40 minutes, is a few opportunities and a few new possibilities at the intersection of close-range human proximity networks relevant for the spread of infectious diseases and respiratory diseases and genetic biological data. So basically what's new is that we have really new technology to measure and map human mobility and proximity at the level of the meters, etc. And we have, as you know better than me, much better, much cheaper technology to sequence both like the DNA of hosts and the DNAs and RNAs of pathogens, right? And so the combination of these two factors potentially changes how we learn about disease spreading and how we design pathogens. So I intend to tell you that close-range proximity is an important source of information if you care about infectious diseases. We know that this is the key driver for spread, for flu, COVID, many infectious diseases that are mediated by droplets we meet while we talk and socially interact. We also know that aerosol is important for the spread of many diseases, and we know now, for example, that for COVID, this was especially important, more important than initially we imagined. And in epidemiology, people have known about the importance of close-range contacts for a long time, and they've used all sorts of old-school technology to map this. This is, for example, a survey from the University of Warwick, where you see people can basically report information on the persons they met, what type of contacts went on. They can even mark the spies. You see, this is a little business matrices, and they can tell, no, Nicolau has met Daniela and put here across, right? So this is very reliable in a way. It provides a lot of context. It's well tested and validated. It doesn't scale very easy. I mean, it's cheap, but you need to convince people to report this information. And also humans have all sorts of limitations in the recall of contacts, especially the small duration contacts. Probably at the end of this day, if I ask you who are the people you spend most time with, you will remember at this workshop, but you might not remember of the many people you briefly encountered in public transport when I'm here, right? And you will know them. So even if you remember an encounter, you cannot link it to that information. Of course, there is a huge potential for smartphones to reach this, because right now everybody else has got basically a tracking device in our pockets, and you saw, and we also, what was the potential of this during COVID when we deployed digital contact tracing strategies that were based on smartphones. I don't know how successful they were here. In Italy, they were not very successful. We had like big challenges in terms of acceptance and compliance with digital contact tracing. And clearly there is a lot more that we have to do to deploy digital means to combat disease, right? The other challenge with smartphones is that as time passes and the technology becomes cheaper, you might have noticed that the cost of smartphones is staying high, right? And this makes sense from the market perspective, because the more the technology is cheaper, the more smartphone designers put more technology into a smartphone. They give you, better screen, faster CPU, and they keep the cost more or less at the same level, right? This is a challenge because in many contexts, you cannot afford to give smartphones to everybody, right? Imagine if you want to study infectious disease transmission in middle-income countries, or if you want to study infectious disease transmission in schools, where now, in many cases, we have policies that actually forbid the use of smartphones because they interfere with learning objectives. So smartphones are powerful. There is a lot more to do there. They also have a lot of limitations, which is why in many studies at large scale on human mobility and proximity, people use sensors. So you use cheap, really cheap, much cheaper than smartphones, custom hardware that does only one thing, and the one thing it does is to measure proximity at the scales of the meters and the seconds, so that you can assess human contacts and close range proximity relevant for the potential spread of respiratory diseases. There are many different technologies available now to do this. I will mention more a technology we use, but this is in no way a limitation on the potential of this. All of them basically come with the same set of hardware, which is basically a small CPU, a small computer, a radio, some memory on board, and some other sensors. For example, it's important in many cases to know whether the sensor is being warm correctly, and so having an accelerometer allows you to know if the sensor is on a person, and what that person maybe is doing, whether sitting or moving around. And the technology is simple. Basically, you need to convince people to wear sensors. This is the difficult part. And once you do, these sensors try to talk to each other, and whenever they're close enough, they can talk to each other, and then they can report this information to an external computer system, or they can save it on board, and then you can extract it after the fact. And this is what the data looks like. This is a workshop. We've been doing this for a long time. So you see, this is back in 2009. This is a workshop similar to this. Right now, people, every point there, every yellow point is a person. Two persons are connected by a link if the system detects them in close range, face-to-face, in this case, interaction. And so you see, many of them probably are looking at the speaker, few of them are chatting to each other, and then the lunch break arrives, and you see social people stand up and social interactions flare up, and eventually people here go to the coffee break, right? So this is just to give you a sense of how this data look like. Basically, these are high resolution, time resolved, close range, human proximity networks, right? And of course, one of the possibilities is to use this data in hospitals, in schools, in many contexts where this is known to be relevant for disease spread, to better understand disease spreading, respiratory disease spreading, and to design computationally strategies for mitigation of disease spread. So the goal is really not to create situations of like surveillance where everybody is tracked all of the time. The goal is to deploy this technology in a controlled, responsible way in a few contexts where we learn, and then what we learn, we can use it in the general setting, right? So we've been doing this for a long time. My team has been doing this for almost, actually it's 17 years now. 45, 50 different places all over the world. And our goal is really to sort of create an atlas of human mobility. So we wanted to learn what generalizes. And to understand what generalizes, you have to look at many schools, you have to look at many hospitals, you have to look at many conferences, so that you can start to see the patterns, the regularities that are relevant for mathematical models. And so, if you want to play with the data are open. We make a point of releasing this data with the public, so that not just us, but the whole research community can work with this data. And this is just an example, but there are like 50, 60 data sets comprising schools, conferences, hospitals. We care a lot about hospitals and schools for obvious reason for you, right? Because these are contexts where we know that the spread of respiratory diseases is important at the community level. We also know that these are contexts, especially hospitals and tertiary care homes or nursing homes, where there are vulnerable people that we want to protect from disease. So understanding exposure and reducing exposure to disease in this context is important. So let me just give you, a flash, since this is a general discussion about how the data look like. This is one of the first things that we did. This was the Science Gallery in Dublin, in Ireland. And this was, it started more as a game. Basically, the sensor people were wearing had a blinking light, and the blinking light was green if you were healthy, and red if you were infected. And this was a way for us to make people, aware of spreading dynamics. So during the middle of the morning, one of the employees of this museum entered the venue with an infected device, and this infected flag was propagated through the day in the conference, in this the museum. And basically, people could see in real time if they were infected or not, and the social dynamics was very fun because you had kids running around and infecting random strangers. You had kids running away from potential infectors. Then this was unfortunately during the H1N1 2009 pandemic started, and then this problem became very popular, which was of course a challenge for public health. For us running this exercise, this was great because 30,000 people went to visit this exhibition because it was very present in the news. We got featured in Science, and so this allowed us to collect really rich data and understand how in a social gathering venue like a museum disease is potentially spread, and mapping how the flow of people, the stationing of people in those places interfere. Just one example. Let's have a look at the school. The school looks like this. This is a primary school in France, and here you see we are in the middle of the morning. In some of the classes you have a traditional kind of lecture. In other classes like that one, you have probably more social kind of learning experience. The school has got 5 grades. Each grade has got 2 classes, so 10 classes. You see every circle there is a class. The point at the center is the teacher of that class. If you don't see the point, it's because that teacher did not consent to wear the sensor. All of our studies, this is important to remark, are purely opt-in. We don't create conditions that are, in a sense, extractive of data or coercive of people. So we try to convince people that that they want to donate data for science, to improve things, but we don't force. People are also bigger, right? So yeah, and then lunch break arrives, and you see what happens. All of the classes become fire up, socially, and then in the school, half of the classes go to the cafeteria, the other half goes to the playground, and then they switch, and then they go back to class, right? And so you can see that these data are complex, right? Because you have multi-level community structures. There is the class, there is the group of classes that goes to the cafeteria together, there are the teachers. Some kids jump between classes according to different activities. So it's really complex. And looking at this data is a challenge in its own merit. And nowadays, as Daniela was saying, we are starting to use more and more machine learning and artificial intelligence to extract signal out of this data. Also, summarizing this data is a challenge because in many cases, this data is too rich for what you care about. Right? And so you want to simply remove information and simplify it by retaining the important stuff. Let me show you one example. If you are a social scientist studying, for example, homophily or gender homophily, in schools, you want to project away the temporal dimensions, and you end up with this. This is the same data, but time is no longer there, and the nodes are colored by gender. And what you see here is interesting, right? And social scientists know about this. When kids are very young, you see that they mix relatively irrespective of gender. But you see what happens when kids grow up, you have a separation of genders, right? So this is before puberty, right? So kids segregate by gender, and then if you do this in high schools, you see the opposite, when the different genders come together again, right? But this is important because basically knowing this also allows you to do better models of relevant for many things. And in this case, actually, the study was a study on mental health of children and how isolation in the social network relates to self-esteem and depression. And we found something interesting. We found, for example, that whereas for girls, being isolated in this network means typically having mental health issues, for the boys, you cannot see it from the networks. So apparently, us boys are very good at faking a social life, but even when we have, mental health issues. And of course, this means that you have to be careful about what kind of signals you use to drive what interventions, right? Just one example, although most of my examples would be about infectious diseases. Now let's look at an hospital. This is Italy's largest children's hospital. It's called Bambino Gesù Hospital in Rome. In this work, they do mostly respiratory infections. So children here come typically with pneumonia. The policy of the hospital is to allow only one parent, one caregiver per children. And here what you see is that this is what people call a role structure population. The nodes, the individuals have specific roles. You have nurses, you have doctors, you have auxiliaries, and you see you have children, and every children is connected with a strong link to the corresponding parent, right? So the color of the link here is related to the temporal properties of the link. A link which is blue, greenish blue, is a link that fluctuates over time, on and off, whereas the orange links are very stable over time, and the thickness of the link is how much time those two persons spend in proximity. And so you see that basically you see these very strong, selective, stable connections between each children and the corresponding parent, of course. And then you see that the rest of the connection to the healthcare system are more like short-lived contacts, right? Doctors coming in and out, nurses coming in and out. You see that nurses interact a lot with one another. Doctors normally interact with nurses who then relay specific tasks to auxiliaries. You see that doctors don't interact with auxiliaries directly. Doctors in general in this hospital are not much in the hospital. And some of them were not very happy to see this data in a sense. We also found, when you do this, you also find anomalies. For example, there was a group of people doing something that made no sense. And they were a mix of doctors and nurses and auxiliaries, for which, when we show the data, everybody was like, this should not happen. And then, we actually, in these cases, you have to work with unions, right? So we promised the unions that we could not link and would not link this information with any personal information on the doctors, right? So we had no way of understanding who those people were. But then when they showed this picture to several people at the hospital, somebody raised their hand and they were like, well, this is the group of people who go out smoking. And which is forbidden in that world, but some people, even doctors actually, they have a small group and they go out and they have a cigarette and then they come back, right? And of course, this potentially has got implications for isolation and transmission. But of course, it also highlights the challenges of materializing this data, right? Because in many cases, people are like, let's collect all the data and then we input things. And very soon you realize that some of the data you collect actually creates liabilities potentially and creates problems totally about your environments. And so you have to be, very, 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 very nuanced and very careful in measuring things. And you have to be many safeguards involved because even if nobody has got nothing to hide, you can still expose sensitive information, right? So just to give an example, as I told you, we share this data with the public, so we publish about 120 papers on this stuff. The community around us publish more than 2,000 papers on this. And so we had the opportunity to map how the community uses this data, not just us. And we see basically three main groups of area, of activity. One is the use of this data for data analysis and machine learning algorithms. This data is very rich, as I told you, and so they can support investigation in data science and artificial intelligence in how to extract meaning from this data, how to extract the classes, how to extract the lunch breaks, how to parameterize this type of data. But also, studying dynamical processes on these networks, dynamical processes on time-varying networks, and so on and so forth. Then there are a lot of social scientists and computational social scientists using this data. Basically, everybody who has been traditionally using social network data is interested in using this higher resolution, more fine-grained data. And then there is the entire community of public health and computational epidemiology. In general, infectious disease modeling and forecast, where this data are used to simulate the spread of diseases, to design in simulation strategies for the mitigation of disease spreading for respiratory diseases. And increasingly, and this is the connection with biology, microbiology, genomics, more and more to do transmission studies, data-intensive transmission studies, where you have simultaneous visibility in the data of the behavior of mobility and proximity, but also on the pathogens that vary in your lab, right? And there is a very rapidly expanding area on the intersection of the social network data with microbiome information, both gut microbiome and oral microbiome. I will show a couple of examples. So let me give you an example of how people in infectious disease modeling normally use this data. Well, the way you use it is like this. You start from the data, for example, in the school, and they provide a landscape over which you can simulate your favorite respiratory disease. And so normally what you do is that you take the empirical data from the sensors and you plug on top of that your favorite disease models. Imagine, for example, a susceptible exposed infectious recurrent model, and then you simulate this in a computer. So the data are real, but the epidemics is simulated, right? And this allows you to do a number of things that normally are not possible. For example, explore, as I was mentioning, mitigation or containment strategies. I mean, just one example. Imagine the school. Normally, to mitigate disease spread, an extreme measure is to close down the school. So one strategy which has been deployed and is part of the policy, for example, in Japan, is instead class selective closure. So instead of closing the entire school, you close a few classes. How do you choose those classes? For how long you shall close them? All of these things can be explored in simulation. And you can do this starting from very simple models. For example, this is a simple study we carried out with a former PhD student of mine, Valerio Gemetto, in Marseille. Basically, this is what happens for a simple like flu-like epidemic as a function of time. If you don't close the school, you see the black curve, and you see this nice epidemic peak. And then if you close it for after you reach a certain threshold for 24 hours, 48 hours, 72 hours. And now we explored another strategy where instead of closing the entire school, when we observe a certain number of symptomatic cases in one class, we close that class only. And we do this for again 42 hours, 24 hours, 48 hours, and so on. And you see that, of course, if you don't do anything, you get the same dynamics. But you see, the nice thing that we discovered here is that a class-selective, reactive closure basically gives you the same mitigation of the entire school closure, right? Of course, this is a very simple exercise. You cannot make any claim that this is viable as a public health response. It's a theoretical model. But then, then you can, the beautiful thing of modeling is that then you can add more and more things. So for example, with Stefano Membella at FPK, we plug this into a much more realistic agent-based model based on time use data that they are in Trento, and there's a place in northeastern Italy. And here is simulated the entire municipality, an entire little town of 100,000 people, where we use a lot of other data, not just our high resolution government networks. And we find similar results. So basically we found that this is a viable interventions to mitigate communities transmission in this simulated town. And then during H1N1, the team of Vittoria Colizza, which is also in the learning project and was mentioned earlier by Daniela, did one step more, and basically they designed, by using the same data, the same school data we collected that I showed you, they designed the screening and vaccination policy to minimize school closure for COVID-19, which was partially adopted by the Italian and French public health protection agency, right? So basically, you see that you start from collecting data, and then you can create, more and more complex, more and more realistic models, all the way to studies that actually have the potential to inform policy and actually do inform policy, right? And as you imagine, during the COVID, especially during the early phase of the COVID pandemic, these data were used a lot, also by us, but by the entire community, to simulate the impact of digital contact tracing interventions, because digital contact tracing relies on this type of proximity data collected by using smartphones. But when we started doing this, we had no data, right? So the only data that we could use were data collected, for example, by our team or by our collaborators by using sensors to try and calculate the parameters of the digital contact tracing interventions. On that, I have an entire long story that probably I will share in the coming days when we, for the more technical lecture, because deploying digital contact tracing, in a sense, was a great opportunity to understand the limits of public health delivered by using electronic devices. Because if you think about that, digital contact tracing was a public health intervention delivered by using the personal devices of individual citizens, which are tied and controlled not by the citizens, not by public health, but by Google and Apple and the other companies. Right? And so in a sense, you have to align three different things, like the needs of public health to mitigate disease, the rights of citizens to privacy, and the business model of the companies running those smartphones. Right? And these three things are very difficult to align. And I was part of the team that designed what is now called as DP13, which is the centralized privacy preserving algorithm that was eventually adopted by Google and Apple. And I believe that in that case we were able to find a good alignment of these three forces. But we also learned that this alignment is not a given, that you have to, that we have to do more so that we can more systematically, more responsibly deliver these interventions in the future. And unfortunately, I believe that after the COVID experience, we are now worse positioned than before in terms of delivering digital contact tracing because of the many controversies and fake news and misinformation around both COVID and digital contact tracing. Right? So I think that collectively there is much more that we need to do to advance things, and unfortunately, like we are all technical persons. Unfortunately, the big problems are not the technical ones. The technical ones are pretty much figured out. We understand them. The big problems are communication and political problems. So we have to also engage those stakeholders. Okay, let me shift gears a little bit. These data are also useful to investigate carriage and transmission of both pathogen and non-pathogen organisms. Right? As we go around, we have, as you know, an entire community of microbes in our mouths, in our guts. These communities do not exist in isolation, right? And we know that microbiome is is potentially relevant for several health conditions, including lifestyle diseases like diabetes and obesity. But we also know almost nothing about the social dimension of the microbiome, right? How similar is microbiome to that, of my colleagues with whom I share an office every day? How similar it is to yours? How similar it is to random people chosen on the street? Like if we place two of us together in a room for 1 day, 1 month, 1 year, what happens to our microbiomes? Do they line or not? All of this is unknown. And so, from a research perspective, it's really exciting. And I want to, share not just what we did, just share what are directions for research and new things that our community together can explore. No? And of course, again, in this COVID was really a great trigger because it lowered the barrier to experimenting with new things. Both in transmission studies of viruses and in other topics. So I will start showing you a few examples of transmission studies where we care about the transmission of something like COVID or flu, and then I will close speaking of microbiome. So this is the plan for the remaining 10 minutes. So a great lab for transmission studies were during COVID these cruises to nowhere. You might remember that some countries, in Hong Kong particularly, they had cruise ships that were going nowhere. They were coming back, right? And these environments were really closed, and since the cruise owners wanted to maintain safety, they basically forced everybody on board, conditioned everybody participating in the cruise to wear a proximity sensor, right? And so this data were used, for example, by the team of Adam Kucharskis at the London School of Hygiene and Tropical Medicine to study COVID transmission in a cruise ship, which is a great isolated environment, right? It's a fantastic lab because these people, by definition, have no contacts with the outside world. And of course, you had at some point an infection cascade started, but we had full information, almost full information on this because people were being tested regularly and people were wearing a proximity sensor. So this was really the combination of rapid COVID tests and proximity information from wearable devices. These wearable devices are pretty bad in terms of technology, but they're also very cheap. And so basically, you see, this allowed this team to create this map of who met whom in the entire community, which is large, our cruise ship. And you see, you have people in entertainment, people in gaming, people in the hotel, the security of the hotel, the housekeeping, passengers. There is an entire community, and of course, what happens is that there are clusters of transmission. Right? So, for example, one of the things they discovered is that the close quarters of the staff, where the density of people is much higher than the density of passengers, actually creates a massive opportunity for spread of disease that then spreads to the passengers, right? And so again, a very interesting lab, and they were able to study and learn about transmission properties of COVID, how much airport transmission was important with respect to close range proximity transmission, and so on and so forth, right? Another context in which there is an opportunity to do a lot of these studies and in which the outcomes, in a sense, are more important is all the work of nursing houses for elderly people, where of course you want to take care of elderly people and you want to limit their exposure to flu or to RSV or to COVID because they are afraid, because for them flu probably is not dangerous. No? So for example, this is just one example of a study, of a proof of concept study we carried out in a geriatric unit in France. In this case, this was a small community. We only have, you see, less than 40 patients, about 30 nurses, 15 doctors. And this is just one sample of the table. Basically, every row is one person, and time flows horizontally. You see these studies lasted for 12 days. And here, every day, we're doing swabs and nasopharyngeal swabs. and doing virological confirmation of basically PCR amplification and virological confirmation for influenza A and B. And in this case, people were also wearing proximity sensors, right? And so you see transmission in the app. For example, you see there is this nurse here, and he got exposed on day 5 to 1, 2, 3, 4 positive individuals, and then he went on vacation for a couple of days, and then he came back and turned positive himself. Right? And of course, this is not a closed system, right? So we cannot exclude that this guy got infected on this day while he was on vacation. And although, given the incubation period for influenza, it's more likely that this exposure was actually cause of this. This, to give you a sense that these data are powerful in terms of understanding mechanisms of transmission and estimating disease parameters, but also that we need mathematically more complex frameworks to assess causal link. Right? You cannot simply look at correlations like I just did. You need to set up a Bayesian probabilistic framework that allows you to place probabilities on infection events and on the transmissibility of the pathogen. And by doing this, basically we could, map the contacts that in this community, you see nurses, patients, and physicians, the contacts between them that were deemed most at risk of having transmitted the disease. Once you have this information, you can go back to the hospital and tell them, look, these are the dangerous contacts. Can you do anything about that? And in some cases, people tell you, no, I cannot do anything about that because that's an essential contact. I cannot remove it. Like a doctor and a nurse interacting, taking care with the patient. In other cases, they tell you, well, this is strictly not necessary. Maybe we can change the cohorting strategies for the nurses. Maybe we can rearrange some of the spaces of the care home so that we reduce this type of contacts, right? So the way we see this going forward is really a process where you measure some things, you show them to the experts, you try to design interventions, and then again you measure. And by iterating over this and by looking at your outcomes, hopefully you can understand, learn, but also improve clinical outcomes. On this, the most ambitious study, we just completed it, so this is a preview for you. It's not out yet, not even as a preprint. This is really a data-intensive transmission study that we carried out in a primary school in Switzerland in collaboration with Lucas Fenner at the University of Bern. What we did here was to use proximity sensors for the kids, for the students in 4 classes in this school in Bern. But we were also doing every day, every second day, we are doing nasopharyngeal swabs, PCR amplification, virological confirmation for influenza A, B, RSV, COVID, and 4 or 5 other viruses. Okay? So basically, we had who has what virus as a function of time for, in this case, 4 months continuously. These 4 months were chosen on top of the forecast epidemic peak for flu, maybe. And then we are combining this, basically the virological data with the clinical data on symptoms from the kids, with the absentee information from the school registry, and with the close range proximity contacts. So this is in a sense the shape of future studies to come, right? Where you have inherently multidisciplinary studies, and you need a team of people with very different competencies that combine the virological and genomic part. In this case, we have the genomic sequences of the pathogens, so that we can really fingerprint transmission. It's not just that I know that I have influenza B and Nicola has influenza B. We do genotyping, so we can link really the virus. This thing just died. Okay, no problem. Basically, you combine this type of rich, like clinical, genomic, virological data with behavioral data, who is absent or not, and with behavioral data of contact, who is in proximity of whom, which people share which class. In this case, we also added CO2 sensors to the classes so that we can measure the level of CO2 in the class and understand if people are opening the windows or not. So in some classes they never open the window and so the air is very stale and you see the CO2 level going up. So we have a proxy in the data for the behavior of over-ventilating the class. And of course we know that for COVID, ventilation is probably even more important than wearing masks at close range in school settings, right? But basically, the goal of the study was really to try and be more quantitative about these things, like how much is important mask wearing with respect to ventilation, with respect to restricting contacts and social distances. The reality is that each of these behaviors got, a risk, and if properly taken care of is a mitigation measure. But what is the weight, the relative weight of these things is something that we can learn from the data. I will not bore you with the data analysis. The analysis of this is quite technical, and we use a relatively new technique called pairwise survival analysis. Basically, we mine from the data pairs of individuals who are equal in all ways except for having been exposed to each other at close range or not. And we ended up discovering basically that in this specific school setting, being in the same class for one day is equivalent in terms of exposure risk to having one short-range contact face-to-face lasting more than 15 minutes. So this is roughly, the equivalence in terms of the risk. And we also discovered that about 60%, no, 55% of our transmission cases were due to sharing a non-ventilated class, and the rest was due to close range proximity. And so you see the picture is complex, right? Like sharing the air in a non-ventilated room amounts to half of the transmission, and the rest is close range proximity. So both are like viable mitigation strategies. And of course this depends on the virus. This I just shared is true for COVID. For other viruses, for example, the short-range contacts are much more important. For example, influenza. Of course, for us this was a sort of initial study. We wanted to see whether this would work or not. And now, like everybody research, we are asking for money to do something larger. But we know that this potentially can work. We have done a lot of these sorts in low and middle income countries. This is why sensors are important, right? You cannot use smartphones easily in environments such as this. This is a research study we carried out in households in rural Kenya. We engaged the local community, like our basically co-authors and field partners who are members of the community. And so, as I told you, our studies are all opt-in. So, we talk, to people in those communities, to the doctors, to the parents, to the kids. We engaged really the community into this data collection, and this was done in parallel in a township, in a normal setting, in a rural setting, so we could understand whether transmission, in this case especially of RSV, was more important, was different in rural settings than in urban settings. We have carried out studies like this in South Africa. This is a collaboration with Cherico and collaborators at the National Institute for Communicable Diseases. And these start to be large studies. Let me just show you this. Here we have 600 people, 120 households. Every row there is one household. Each of these waves lasted 1 month. So basically we are looking at a study that lasted almost 2 years and tracked these 600 individuals. And you see for some waves we take data asynchronously across households, but for some waves we made the effort of activating data collection in all of the households at the same time, so that we could also map transmission between households. This was mostly on COVID, And I would love to say that we learned a lot, but actually it was a very difficult study to run. We haven't learned much. We basically learned, the one thing we learned in the study was that sharing a household was much more important than close relatives. For COVID, it makes sense, right? And then, there are more nuances result, but this is a general lecture, so I just want to give you a sense of things. And finally, I promise you something about microbiome, right? So let's go back to a school. This is one example, just one example. This is one school, this time in Norway, and it's a primary school, a small one. We only have 50 kids, these are 2 classes in this school. And for 4 months, every month we had 1 week where kids were wearing sensors, and 2 times a week, as part of their school activities, these are Norwegian kids, they're very disciplined. The kids themselves were splitting in a vial and then processing the sample. And so we had twice a week microbiome information about their total microbiome together with the comments, right? So basically, we have the proximity network from the sensors, and we have the oral microbiome from the saliva samples. In this case, since we have very little budget for the study, we use the simplest technology we could, and as you know, it's very limited, so we use plus 16S to characterize the bacteria, the microbiome in the mouth of these kids. And so we have basically, for every node, we have a phylogenetic tree of the bacteria in the mouth. I'm a physicist, so I don't know a lot about microbiome. I was shocked to learn that 80% of the bacteria in our mouth, we don't know what they are because we cannot capture them. And but basically, we could track about 600 species of bacteria in this community. And one cool thing that we could do is that based on the microbiome information in your mouth, we can predict who are your friends. So if everybody of us would give me data on their microbiome, I can predict who is friend of whom in this community. Just because of that, right? Which is super interesting. And of course, this opens more questions than it closes. Because in a sense, does this mean that the oral microbiome is transmissible? Yes or no? We don't know. The reality is that we don't know. Were these kids already aligned in their microbiome because they had spent time together? Was the microbiome already equilibrium or not? We don't know. We also tried to run this study across Christmas, so kids would go home and then come back. We are hoping, the things would get different. And no, apparently the immune system is very good at keeping in check your microbiome. Your microbiome is yours. But there is a component that's similar to your closest friends. So it's my point, we have very complex data and homophily things. Maybe I have friends with Nicola. And so this correlates with the fact that we eat similar food, and then this influences the similarity of our microbiome. So these factors are certainly at play. But there is also evidence in the literature for genuine microbiome transmission. Right? And some of the bacteria that we found transmissible are responsible for cavities and for gum disease. And so potentially there is a social dimension to these diseases. Right? This also matches much better study than ours. For example, let me just close with this one. This is an amazing study by the team of Nicola Christakis at Yale University, where it's similar to the case of the isolated cruise. In this case, they had isolated villages, and where they were focusing on gut microbiome rather than oral microbiome. And they were not using sensors, they were interviewing people and asking them about their familiar and friendship relationships, right? So this is self-reported data on the families. And surprisingly, also in this case, they can predict exactly in the way we do who is friends with whom based on the similarity of the intestinal microbes, right? in this case, and they can do much more. So if you're interested in this, I invite you to read this paper because it's amazing and they open an entire field of research. But all of this to tell you that basically there is a massive amount of work to be done at the intersection of quantifying social networks and looking deeply via genetics at the bacteria in our bodies. And we have to develop new methods. We have to develop new methods mostly because these genetic, let's say, omics data that we have are super highly dimensional, and so increasingly there is an opportunity and a necessity to use AI techniques to do dimensionality reduction and to make sense of this type of data. I will close with this. Thank you so much for your attention.
I think we have some time for some questions now.
Hi.
Yeah.
According to the microbiome, you can trace the friendship between children. I just wonder, between friendship and family, like parent and children, can you track that as well?
That's a great question. In our case, we cannot because we are only collecting this data at the school. But in a sense, this was by design because we wanted to run this study in a community of non-cohabiting individuals. Because in a sense, it's already known that between cohabiting individuals, there is some sort of alignment of the microbiome. Our question was, if you take a group of people who do not live together, who do not share the same food, the same environments, who do not breathe the same air at home, can we find a signal of correlation between microbiome and social network structure? And of course, these are primary school children, so it's expected that probably, they put their hands in their mouth and they share a lot of fluids between them. And in the sense, probably, there are mechanisms for passing each other bacteria. But how much transmission is possible and lasting in this context are open. Another big question we have is what are the time scales? These are completely unknown right now. So one of the things we want to do is to run this study at the very beginning of the school year, so these kids have never met before. And then we see whether their microbiomes align. Right? Again, this is another question. And what I love about this field right now is that nobody knows even the basic things. So and genetics and genomics are really mature for this. Right? Paradoxically, the behavioral data, the sensor, the proximity sensor data, is trivial, simple. And the real challenge is to put in a network context the genomic data, but technically, in terms of data analysis, doing data analysis of genomic data on a graph is very complex. Because we are potentially all sort of confounders, and we have to use really sophisticated methods to do data analysis and assess actually transmission, causation, and so on and so forth.
And do you have any more questions? I actually have several. Yeah.
Hello, thank you very much for your nice presentation. I'm Silva. I'm from the Marketplace Corporation. According to the concept, especially for proximity, I'm very interested if I would like to apply this idea with the non-communicable disease. Is it possible to do that? Thank you.
Yeah, there is quite a rich literature on using the social network structure as a background for studying lifestyle diseases. Particularly, there is quite a strong literature on diabetes and obesity. And actually, the team of Nicholas Studies carried out a seminal study called the Framingham study, that ran for almost 20 years, where they were mapping social networks of people and the series of clinical outcomes. And of course, what happens is that in a social network, all outcomes are correlated with the networks. If not else, because your friends are, if you are rich, your friends are richer, right? And rich people tend to have fewer diseases. And so now you're correlating on that, right? You're more healthy, but just because like they're all caused by socioeconomic determinants, which however are correlated in the social network. But people have investigated this, and actually there is an ongoing debate, for example, on whether obesity is actually more transmissible than it seems, not because of biological reasons, but because of behavioral reasons. Maybe there are behaviors that are contagious along the social networks, and these behaviors translate into health-related, into risk behaviors for chronic diseases and lifestyle diseases. So, yes, there is a lot of literature and a lot more to do on this going forward. People have used all sorts of social networks, also online social networks, data from Facebook, data from Twitter, to understand the interplay of clinical outcomes and social network structure. But the studies are very complex because not only diseases, well, let's say, even for non-communicable diseases, behaviors and cultures transmit over the network, right? Because information always transmits over the network. And so this creates very complex situations that are very difficult to understand, but it's, that's why it's interesting, so yes.
Do you have any more questions? I mean, I have some questions. So it's very interesting, and because you mentioned about like, agent-based information of about 100,000 agents, what sort of computer do we need in order to do such an analysis? Do we have like, to use HPC or workstation will be sufficient?
Like for a lot of this work, computation is not a bottleneck, not a significant bottleneck. For this type of simulations nowadays, we can run most of them on laptop at the individual level. Where things become complex, but I believe that Nicola in particular will address some of these aspects, is when you do parameter estimation or when you have, in a sense, to compute like confidence intervals. At that point, you have to sample not just the scenario that you are investigating, but all parametric variation by sampling the parameters according to distributions that are your best representation of the knowledge on those parameters. You end up using, macro chain Monte Carlo techniques or other sort of parameter sampling techniques. Increasingly, there is an interest in using AI to optimize sampling strategies, so that you can do potentially, and there are already it's better than macro chain Monte Carlo, and it's smarter. You can use fewer points. But if you start doing that, then what basically becomes heavy is running your individual simulation for these many parameter combinations. In that case, what you do is that you typically deploy in the cloud and you have to create a large number of instances. And of course, like we use more and more AI. The moment you touch AI, you need GPUs, and then suddenly your computational requirements and costs So there are potentially HPC aspects into this. They tend to be simple because a lot of the computation is trivially parameterized, right? So normally you scale very nicely. So you don't need, in a sense, like if you have to talk to a proper HPC person, they will tell you that yes, it's HPC, but in the simplest possible way. And that's fine with that.
On the topic of model fitting, which is very interesting because we learn from AI, is modeling that without data, modeling or simulation is just a theoretical exercise. And we need to use data to evolve our model in order for them to be useful. So I just wanted to know, in terms of the data. So you mentioned that you showed that in many cases you get 90% compliance, lots of participants. But in general, for example, in a hospital, what sort of percentage of recruited participants to join? Because at the end of the day, as you showed, this close proximity network information is very personal and it can lead to
Yeah. So that's a great question and it's complex. I mean, it is like the level of coverage of the community you have is critical, right? And again, think of the example of digital contact tracing. That only works if you have very high participation in the intervention, because basically your coverage of the connections is quadratic in the coverage of the people. Right? So if you cover half of the people, 0.5, your coverage of the potential connections is 0.5 squared, so it's 0.25. So only 1/4, right? And actually it's worse because of the heterogeneous nature of social networks. So basically, it's critical to have high coverage, especially if you want to do transmission studies, because otherwise you sparsify the network enormously and you don't see a lot of the connections. At the same time, we want to maintain our studies really maximally ethical and respectful. We don't want to be extractive in the. Already all of the digital platforms are already very extractive for us in a sense. So we want to be very respectful of this. And so all of these studies are opt-in. And also right now there are so many regulations that really basically rightfully limit what you can do even for biomedical studies. So our only strategy is basically to engage, to devote a lot of time in engaging the relevant stakeholders into data collections. So for schools, we normally start from the director of the school and then the teachers, and then we have an event with the parents, and then with the kids. So you really need to involve everybody, because it's very simple to mess up with a study like this, and sometimes we make mistakes, and the community can respond and destroy the experiment in a moment. We had one case where a class, at some point, they had a swap party where they swap all of their sensors. And that was it. They killed our data collection in half an hour, right? So for example, in schools, we make the sensors customizable. Every kid can draw it, they can make theirs, so we are sure that they pick up the right sensors when they go back to class. You have to work a lot of this, and we have made many mistakes. In low and middle income countries, for example, compliance with the local aesthetics was very important. An electronic device, we learned, is regarded as not cool. And so people want to really customize it, use local fabrics so that it can be worn in accordance to the aesthetics they like. For hospitals, some aspects are simpler, because in a hospital you already operate in an environment. First of all, people are aware of the need to be observed in a sense. Of course, in an environment where potentially your body is invading your blood, right? On the other hand, and it's also legally simple because you have frameworks in place for informed consent that you can establish perimeters for sensors to be worn. On the other hand, it's a very, by definition, it's an environment where people are vulnerable and where they might consent more than they should just because they feel vulnerable. And so again, what we do is that we engage the full stack from the hospital managers, the unions, because the doctors, of course, respond to the unionized, at least in Europe, and it's illegal for us to measure presence on the workplace for employees of the hospital, for example. And then we engage the patients and everybody. So normally, in all of the studies I showed you, we have more than 90% of participation. In the studies in Norway and Switzerland, we exceeded 98%. Right? So really, basically in Norway, only one person was not on the system, and in Switzerland, only 2 persons were not on the system. You have to work for this. The other thing we learned is that this thing decays with time, but since we could measure it, mostly because of the on-board accelerometers, we know what are the laws of decaying compliance. And so we know also from the data how much we can push it and when it's useless to push it. Right? And so this gives you a natural bound on the duration of our experiments, which is not initially we thought that the limit were the batteries of the devices. The limit is the patient or people. People after a while, they just come.
What would be the percentage that you consider now is useless? 70%, 80%, 90%?
It depends on the question, right? But I would say if you think of transmission studies, 75-80% is the limit. And you need to be above that. If you are looking at other types of cheaper interventions, then 50% might be enough. For most study in sociology, where we try to put the individual in the context of only their neighbors, you can get results with 40%, 30%. But I would say it's difficult to say in general. It depends a lot. But normally the answer is hard. So normally you have to work for hard. Which is also why, probably the strategy is to work hard in a few contexts, learn as much as possible, rather than do many studies at lower components. Right? So a single study done right, gives us much more in terms of knowledge.
Thank you.
Thanks, guys.
That's all.